Chapter 5, Other Catabolic Pathways. In this topic, the objectives are to compare the pentose phosphate pathway and the Enter-Duerhoff pathway with glycolysis in terms of energy production and products, describe fermentation, and contrast it with respiration. List 3 useful end products of fermentation and explain how fermentation reactions are used to identify bacteria. Explain how lipids are catabolized for energy and metabolite production, and explain how proteins are catabolized for energy and metabolite production. We're going to be covering the alternatives to glycolysis, which are the Enter-Duerhoff pathway and the pentose phosphate pathway. Then we're going to talk about fermentation, lipid catabolism, and protein catabolism. The textbook starts out with the pentose phosphate pathway, but I find it better to start out with the Enter-Duerhoff pathway because it's closest to glycolysis. So we take a look up here and we start with glucose. Just like we do in glycolysis, we take an ATP and through substrate level phosphorylation end up with glucose 6-phosphate. Here's where it changes. We bring in a new electron carrier for a redox reaction, which is going to happen here. It's a cousin to NAD+, it's NADP+, and it's used in anabolic reactions. NADH is generally used in catabolic reactions. It's part of how the cell regulates the two different pathways. So when the cell needs to generate some NADPH for catabolic reactions, then it uses the Enter-Duerhoff pathway. So we get a different product here, we split, and we end up with our old friend G3P. Now this goes through the rest of the steps of glycolysis and goes on to the Krebs cycle. But we only form one of them. We only form one NADH and 2 ATP instead of 2 NADH and 4 ATP like we would in glycolysis. Now the other half of this molecule is pyruvic acid, and this can go straight to the Krebs cycle or to fermentation, which we're going to be talking about next. So the thing to keep in mind when looking at this pathway is to look at how these products differ from the products of glycolysis. So we end up with G3P, just one of them, and a pyruvic acid. We end up with consuming just one ATP. We end up producing one NADPH, one NADH, and 2 ATPs. So we have 1 ATP net production. On to the pentose phosphate pathway. I'm sorry, this is a mess. We take glucose, we add ATP to it, we generate an NADPH, which go on to anabolic reactions. We generate some more here. So part of the goal of the pentose phosphate pathway is to generate NADPH for anabolic reactions that have redox reactions, or in other words, require electron donors. Then we come up with this funky 5-carbon sugar called ribulose 6-phosphate. This is necessary for the Calvin-Benson cycle in photosynthesis. So if you have a photosynthetic organism, they're going to need to have the pentose phosphate pathway so that they can produce this ribulose 5-phosphate. We can, if we don't need that currently for the Calvin-Benson cycle, or if it is not a photosynthetic bacterium, we can switch this around and we come up with xylulose 5-phosphate and ribose 5-phosphate. Ribose 5-phosphate goes on to make nucleotides, which is a good thing. You need to have intermediates to make your nucleotides so that you can synthesize DNA and RNA. So we can take these and we can mess around with them a bit. We can make some G3P, which goes off to glycolysis, so we can generate energy from this process. Remember from my example of feedback inhibition, here we have erythrose 4-phosphate, which is a 4-carbon sugar, which goes on to pathways that synthesize amino acids. Then, if we don't need to be synthesizing amino acids, if we have need for other stuff, we can mess around with this and make fructose 6-phosphate, which goes on to glycolysis, or glucose 6-phosphate, goes into glycolysis, or it can come back up here if need be and then we have our friend G3P. So like I said, this is a mess, but here's what I want you to get out of it. The goal of the pentose phosphate pathway is to generate intermediates for these other pathways. If we don't need the intermediates for these other pathways, we can convert them back into molecules that can go on to catabolic pathways that generate energy. So it really is kind of cool, and it makes it so that the cell is self-sustaining. If you don't have the pentose phosphate pathway, suppose you can't produce ribose 5-phosphate, then you have to bring in those intermediate precursors bring it in from the outside of the cell, bring it in by eating it. So you have to rely on another organism to make those intermediates. For organisms that have this pathway, they don't have to rely on other organisms. So once again, make sure you know how these products differ from the products of glycolysis. On to fermentation. So if we look at our overall chart of catabolic pathways, respiration, we go through glycolysis, we go to acetyl-CoA formation, the Krebs cycle, and then onto the electron transport chain. We're going to go off on this side road. So suppose that our organism is cut off from oxygen and the electron transport chain. We can't pass on those electrons, so this whole system shuts down. We don't go through the Krebs cycle. What we do instead, if you are a microbe or your muscle cells, going with a multicellular example that you're already familiar with, we can go into fermentation. The reason why we go into fermentation is so that we can maintain glycolysis, maintain this metabolic pathway so that we can get those 2 ATPs. Because 2 ATPs are better than no ATPs. Even though respiration generates something more like 38 if you're a prokaryote and 36 if you are a eukaryote. So let's take a look at fermentation. So we take the end product of glycolysis, pyruvic acid, and let's go with your muscle cells. So what we do is we take this NADH, it's not going to the electron transport chain. We need NAD plus to keep glycolysis going. Remember, we used an NADH right after we split our 6-carbon molecule. I was going to say glucose, but it's not glucose at that point. We need an NAD plus. So we take the NADH, take the hydrogen off, put it onto the pyruvic acid right here, and make lactic acid. So this NAD plus can go back up to glycolysis, and we can continue to get those 2 net ATP. Since we're talking about your muscle cells, when you start doing heavy exercise like running or lifting stuff, it takes a bit for your respiratory system to get caught up to the oxygen needs of your muscles. So your muscle cells temporarily switch over to fermentation, produce lactic acid, and that produces the burn in your muscles. Once you start getting respiration going again, we take lactic acid, we pick up a hydrogen to make NADH, turn it back into pyruvic acid, goes back into the rest of respiration. Cool, huh? Now, let's use a microbial example, because, this is microbiology. We have microbes that do not have an electron transport chain. They do not undergo respiration. So they are limited to fermentation. Now, one of these bacteria that we have domesticated, we use it a lot, are lactic acid producing bacteria, either lactobacillus or lactococcus. They undergo this pathway because they do not have an electron transport chain. So they have to do fermentation. So they take pyruvic acid, do the same thing that your muscle cells do, and produce lactic acid. And what does the lactic acid do in the milk that the lactic acid producing bacteria are producing? Well, it curdles it. And we make cheese out of it, and sour cream, and all sorts of other wonderful things like yogurt. Aren't you glad for lactic acid bacteria? Also, lactic acid producing bacteria are part of your normal microbiota. We have them in our gut. If you're a female, you have them in your reproductive tract, And by producing the lactic acid and lowering the pH, it makes an environment that is not suitable for some pathogens. They're there to protect you. So we keep them moist and warm, and we feed them, and they protect us from other bad bacteria. Aren't they just wonderful? Now, let's talk about this other pathway. Some yeast, Saccharomyces being a main example, do not undergo lactic acid fermentation. Instead, they take the pyruvic acid and they knock off this acid group. So they produce CO2. So we end up with acetaldehyde. Once again, the whole purpose of fermentation is to generate NAD+, so that we can undergo glycolysis. So we take this H, put it on right here, and we get ethanol. So this is why alcoholic products are always bubbly, because they're producing CO2. We do not have bubbly yogurt, because lactic acid producing bacteria do not clip off this acid group here. Sometimes what the product we're after is ethanol. Sometimes the product we're after is the CO2. Saccharomyces is also used to make bread. They do have an electron transport chain, so when you're making bread, you start them out with sugar in air, and they grow really fast because they're producing a lot of CO2. Then you take that increased population of yeast, mix it into your dough, and you cut them off from oxygen. So they have to switch over to fermentation. So they start producing this gas, which causes your bread to rise, which is how we get nice, fluffy bread instead of tortillas. Tortillas are wonderful. Fluffy bread is wonderful too. So when you bake it, you burn off the ethanol. Those are two fermentation pathways. Other bacteria and other fungi produce other products. So Escherichia and Acetobacter produce acetic acid, i.e. vinegar. We have Clostridium that produces acetone and isopropanol. We can produce this in the lab, but bacteria are far more efficient at doing it. Here's our friend Saccharomyces. They produce CO2 and ethanol. Aspergillus is used in making soy sauce. Lactobacillus, Streptococcus, they help us with our dairy products. Propionibacterium produce CO2 and propionic acid. That's used as a secondary culture to make the holes in Swiss cheese. Let's talk briefly about identification before I go on to breaking down stuff other than carbohydrates. We can identify bacteria by their fermentation products. Because if something is producing acetic acid, we know it's not Saccharomyces. If it's producing acetone and isopropanol, we know that it's Clostridium and not Escherichia. Kind of see how that works? In the lab, we will be doing some tests that show whether an organism can ferment a carbohydrate or not, and looking at the byproducts of that fermentation. It's part of bacterial ID. Now, to this point, we've made it sound like the only thing that you can get energy out of are carbohydrates. But, as you know, we can get energy, or calories, out of lipids and proteins. So we're going to talk about how we do that and how microbes do that. So here is a triglyceride. So we've got 3 fatty acid chains. We've got them attached to glycerol. So we undergo a hydrolytic enzyme with a lipase, and we get glycerol. The glycerol gets changed to DHAP. Do you remember DHAP? When we do that first split of the 6-carbon molecule into 2 3-carbon molecules, DHAP is right there. So this goes on to glycolysis, produces some NADH and ATP. Now we're left with these fatty acids. Well, we undergo a process called beta oxidation. So we mess around here, generate some FADH2s and some NADH, which go onto the electron transport chain and help produce ATPs. And we're left with an acetyl-CoA, which goes onto the Krebs cycle, and a shorter fatty acid chain. So basically, beta oxidation is the process of snipping off 2 carbons at a time that go on to the Krebs cycle. So we get all sorts of energy from lipids because they're bigger molecules and because they have more hydrogens. Each of these carbons has 2 hydrogens on it, well, except for the ones on the end. They have 3. So we can use these high energy hydrogens in oxidation reactions to produce energy. That's why many microbes and why humans do long-term energy storage in the form of lipids, i.e. fats. On to proteins. Now you can get energy out of proteins, as evidenced by the Atkins diet. Proteins are too big to bring in through a cell wall or even through a cell membrane. Let's go with a multicellular example that you're used to. We eat proteins and our digestive tract produces proteases that break down the proteins into amino acids. Then your intestinal cells can take those amino acids, pull them in, eventually ends up in your bloodstream and everybody gets to share. Bacteria and other single-celled organisms do not have a digestive tract, so they release proteases out into the environment, does the same thing as what's happening in your stomach and your gut, clips these amino acids off to make individual amino acids, which are small enough to bring into the cell. Then we clip off the amine group in a process called deamination. This comes off as ammonia most of the time, and we now have something that we can send to the Krebs cycle. We can either use this part that's left as an intermediate to make other amino acids that the cell can't make, or they may need and they're not getting them from the environment, or we can send it to the Krebs cycle to generate energy. Cool, huh? So that's it for this topic. So here are some reminders of what I want you to get out of this. And next we'll talk about catabolic reactions, or the process of making bigger molecules from smaller molecules.